Welcome to our webinar today. We'll talk about how the LeviCell is an ideal tool for live cell enrichment in a variety of different applications and different sample types. Our technology is gentle, simple, and label-free, and it's the only currently available commercial platform for live cell enrichment that does not use labels or specific markers. Now, to start off with, sample processing is actually the most difficult and often quite overlooked part of any downstream analysis. Regardless of the type of starting sample, whether it's blood, biopsy, tissue, cell culture, and regardless of what downstream analysis is being performed on that specific sample of interest, live cell enrichment is a key step that is critically vital for a high quality analysis. The old saying of garbage in, garbage out could not be more true here. If we start off with a complex biopsy or tissue sample where the sample contains large numbers of dead cells or debris, our ability to enrich for live cell population, doing so gently, efficiently, and producing high quality cells is vital to our sequencing reaction, our single cell analysis, or even FACS workflows. Without this critical step in the middle, doesn't matter how sexy or how advanced the analysis method is, we will not be able to achieve the results in the data that we are looking for. Now, this becomes especially true when we look at some of the most difficult primary samples out there. And this is where our platform has been extensively validated by existing customers and really validated to work extremely well with some of the most precious and difficult samples. These include samples that are considered to be low viability. Low viability samples for us means if 50% or fewer of the sample contains viable cells. Great examples of these samples are tumors, frozen samples, archived samples, any sample that has been stored for a long period of time. Quite often, at least 50% of those cells are dead and make that sample very difficult to process. Just as an example of how big this need is, in the US, there's over 100 million of these archived samples waiting to be analyzed today. The next area where we really excel and where our customers find great use for the technology is with low quantity samples. These are samples where typically you have fewer than 1 million viable cells. Things like fine needle aspirates, swabs, biopsies will quite often have anywhere from 10,000 to 100,000 cells. The current methods and technologies, which we will describe on the next slide, fall short of being able to process any sample with fewer than 1 million viable cells. This is a great area of interest because some of the most valuable primary samples, things like biopsies, fall into this category where you have very few viable cells to start off with, and as a result are unable to successfully analyze them, regardless of how advanced your analysis method is. And finally, perhaps the most important sample type are the sensitive cell types. These are typically your primary cells, primary samples, things like macrophages, adipocytes, pancreatic cells, and lung cells, cells that are extremely sensitive to handling, cells that are easily killed or activated by pressure or any force. From our experience, over 70% of primary cells fail processing with standard methods. Either they are killed due to a high pressure, long processing time, or sometimes something as simple as taking them out of their original media is hugely detrimental to their function. Now, when we go out and speak to our customers, there are 3 current technologies we hear that are being used today to different degrees to success. And to go over this quickly, it would be very important to highlight how the LeviCell platform is different and superior to what's currently available. The first and probably the most popular method are magnetic beads. These are usually sold by Miltenny. They are sold in different formats and different sizes. However, universally, they suffer from very poor yields and viabilities. When we go through our comparison data, we'll show you how our system performs versus a standard Miltony prep. Typically, 10 to 20 yields are average, if not exceptional, for Miltony, and quite often, a majority of the sample will be killed or destroyed during the processing. Additionally, beads tend to be very expensive. One must think about the cost of processing beyond just the cost of the reagents. An average bead run takes anywhere from 45 minutes to an hour. It requires quite a bit of hands-on time. And additionally to that, the ability to automate is very difficult due to the large number of steps and the high failure rates. And finally, perhaps the most important point here is that even if beads are the correct choice in terms of your workflow, chances are if you are working with primary cells, beads will very negatively affect the performance and viability of the cells. They either tend to activate cells, such as macrophages, they tend to damage cells, such as lung cells, or are completely incompatible with some cell types where a cell surface marker is not present in order for the bead to bind to. Because keep in mind, in order for beads to work, what is required is a specific cell surface marker to identify the cell type of interest and then hybridize to the bead. And in fact, that is that interaction which allows for the enrichment. So without a cell surface marker, or a poor surface marker, means the beads are not a viable choice for your experiment. The next, and perhaps the most commonly known tool, is a centrifuge. It's been around for hundreds of years and obviously continues to be present in almost every single laboratory worldwide. What makes the centrifuge a bit difficult to use is that as it's a very manual process, requires density gradient, it requires very large numbers of cells to use, usually in the millions. And as we've described in the previous slide, majority of our primary high-value samples never reach those large cell numbers, where a centrifuge would be a reliable means of separation. Additionally, I think all of us who have worked in the lab remember how difficult and painful it is to work with a centrifuge. This is not a simple process. It requires a very experienced operator and quite often has a huge amount of variability from run to run. And finally, cells unfortunately do tend to be killed or activated by the G-forces. In order to get the separation to work well, the cells have to be spun at a fairly high rate, which in fact quite often will activate or kill them. Now moving forward to our final analysis and separation tool, these are FACS systems. These are probably the most commonly known for cell isolation. Unfortunately, they require very high pressure and small nozzles to separate individual cells into droplets. And this high pressure and squeezing force will quite often kill or activate most primary cells. Additionally, these instruments are prohibitively expensive. An average FACS platform these days is about $250,000. And finally, these are really high-tech instruments requiring a full-time experienced operator. These are not instruments that are easily incorporated into everyday workflows with a standard user who just needs to isolate a specific life fraction for a downstream experiment. So now, having given you the background of how the current platforms work, let's go ahead and talk about how we are different. So the LeviCell is really the first commercial platform available which allows the user to isolate specific life fractions of cells without the use of any antibodies or dyes. The system has multiple different capabilities and features that make it very unique and ideally suited for working with rare, precious primary samples. To start off with, we start with a sample in suspension, and our sample in suspension right away is our first differentiator. What makes us different than beads, FACS, or even centrifuge workflows is that we inherently do not care about the size of the particles in suspension or what the suspension is. This means that this exact same workflow can be used for all researchers, regardless whether they're working with very small bacteria or very large organoids, embryos, or small animals. We have no size restrictions in terms of the size. We can also easily work with different liquids. Some customers may choose to start with blood, others may work with cell culture media, and yet another fraction may start with something such as dissociated tissue in PBS. As long as the sample is not so viscous that it cannot be pipetted, in other words, as long as you can pipette the sample into our cartridge and the particles are under 350 microns, the process is exactly the same for all use cases. Now, once we have the sample in suspension, we simply pipette it into the chip. The chip goes into our instrument, and the instrument performs the magic of levitation. Here, what you're seeing is an image obtained by the instrument. We have a camera inside, which contains a bright field and two fluorescent channels. And here we are seeing a green and red channel overlay. Now, a critical point to remember is, even though we're showing you green and red here to identify live cells and dead cells, that analysis is purely for analytical purposes. What I mean by that is, even if the imager and the fluorescent channels were not present, the separation would occur in the exact same fashion. Unlike a fax platform, separation does not require a feedback loop which identifies cells of interest. Here, our imaging is solely for analysis purposes and provides the information in terms of cell number, cell size, and cell behavior. Now, the automatic separation is a little difficult to comprehend at first, but when we go through the technology slides, I think it will make perfect sense. So from here, the automated label-free sorting occurs inside the instrument, and the customer is now able to collect their live cells and dead cells into two separate tubes. The entire process takes under 20 minutes. There is no labeling, there is no waiting, and there's no purifying. It's a fully enclosed process with no additional steps required. Now, what allows us to do some of this is several features of the system, which are ideally suited for live cell enrichment workflow, right? And these include no clogging. Again, our traditional fax, bead, or even centrifuge workflows require pre-enrichment or pre-filtering. Here, we're able to start with any sample with particles under 350 microns. The entire platform is solid state. That means that run to run, there is no need to calibrate or set anything up. In fact, our system-to-system reproducibility is so high that we do not even require a new system installation calibration or recalibration at any point in time. The entire platform uses under 1 psi of pressure, which means even the most sensitive primary cell types are not activated or killed. There's no beads or markers included. And perhaps one of the most beneficial aspects of technology is, for all of the reasons above, we're also able to process dirty, complex samples with no debris removal or pre-filtering required before the sample is added into the system. Now, what makes the entire platform work is our cartridge. This is a very simple system that allows 100% of the sample to be processed, again, with no cleaning, no calibration, or no contamination issues. The sample that we've discussed is pipetted into the lower chamber. It is processed through the chip, and it comes out at the top of the chip into two different collection ports, a live cell and a dead cell collection port. These two ports are then pipetted out into tubes which are ready to use for downstream analysis or processing. Now, the beauty of our device and our consumable is that the volume of the sample always stays 100% of board of the cartridge. What this means is, run to run, there is no concern of cross-contamination. The sample never touches any part of the hardware system. The two little ports that you see up here are vacuum connections. This is how the sample is effectively pulled through the cartridge, and these two ports are completely isolated away from the actual volume of the sample. What this means is that run to run, there's no aerosols, everything stays in liquid form, and there is no cross-contamination because the vacuum ports are completely separate away from the actual sample. The consumable is also irradiated for sterility, which means it's ideally suited for any work that may require downstream cell culture or any sort of PDX model work, for example, where the cells have to be grown or maintained for a longer period of time. Now, finally, before we move away, this consumable is very reproducible. It's a solid state system. All the channels are a fixed size, which means run to run and instrument to instrument. Again, we have a very high reproducibility with no need for calibration because, again, it is a fully solid state platform with no moving parts. Now, before we go into the technology, let me highlight a little bit about what we believe is one of the key bullet points in terms of how the levitation platform can change your workflows. Unlike all other methods on the market today, whether it's fax, beads, or even centrifuge, we do not have to remove any debris before processing. What this really means is when you look at the true workflow for either of the other platforms listed here, the majority of the workflow is upfront. The first 2 hours and about 8 steps are all about pre-processing before the sample can ever go into the instrument. For the LeviCell, we start directly with a dissociated tumor sample, and within 3 manual steps in 20 minutes, we are done. So even though our actual workflow is much easier, in reality, the debris and cleanup removal required by all other methods is the actual game killer. So how does the technology work? Well, as I've mentioned in the past, everything's solid state. If we were to zoom inside the instrument, what you will see is there are 2 rare earth magnets bolted to the top and bottom where the consumable, or the channel in this case, slides in or fits between these magnets. These are our standard rare earth magnets. They are slightly more powerful than what you would attach to your fridge, but in effect no different. The channel itself, in this case, is a 2 millimeter high, 2 millimeter deep consumable, and effectively is just a channel which allows the cells or particles to flow through. Now, if we were to take our cell suspension and place it into liquid between the 2 magnets, there are several forces that are in fact acting on the particle. The first and the most important is what we call the magnetic force. In order to make the entire system work, we spike in a little bit of FDA-approved contrast agent into the solution in which the cells are placed. This is an FDA-approved compound. It is quite often used for a variety of therapeutic applications, as the name implies. And more importantly, is the amount we are using is significantly lower than what's actually injected into the patients. This is a completely inert compound and does not actually affect or bind to the cells. What it does do is it creates a density gradient that effectively follows the magnetic fields. What I mean by that is where the magnetic field is stronger, near the bottom of the channel, then the amount of the MRI contrast agent molecules is greater. As the magnetic field decreases, the number of MRI contrast agent molecules decreases. And so what we have effectively created here is a virtual density gradient where the cell or the particle of interest is fighting for space with the molecules of the MRI contrast agent. So on one hand, the force of gravity is pulling the particle down, and at the same time, the molecules of MRI contrast agent are pushing it up. So if we take a particle of an average density, it will end up somewhere in the middle of the channel because the actual equilibrium height of the particle is directly correlated with its density. So particles of low density will levitate higher. One way of thinking about this whole configuration is that it's very similar to an analytical centrifuge, where the final configuration or where a specific compound will end up in is its molecular weight. How quickly it moves through the density media is a function of its size and shape. And in this particular configuration, we have the exact same thing. The final equilibrium position of any particle is its density. How quickly the particle arrives there, or the speed at which it moves, is a function of its size and shape. And this is what allows us to isolate specific populations based on their morphology. Now we spent talking about live cell separation. So how does that particular function work? Well, if we start off with a dissociated tumor, which contains live cells, dead cells, and debris, Live cells levitate to the top of the channel, dead cells and debris tend to sink. The reason for this is quite simple. Just like most dyes that are used to identify a live cell or a dead cell, a live cell has an intact cell membrane, and it in fact has buoyancy, so it floats to the top. A dead cell, as the cell membrane opens up, the media or the solution in which it's in floods in, and effectively it sinks the cell. So what we're doing here is we are using the inherent stability and the cell membrane functionality to separate out live cells from dead cells. Now, once these cells are up levitated, it's quite easy for us to collect the fraction of interest. Now, to show you the power of levitation and actually show you that in fact live cells levitate and dead cells sink, we have created a video in which a breast cancer cell line is being slowly poisoned. So as you can see, the video starts at 15 minutes. It is slightly accelerated. And over the course of the movie, what you will see is cells that are starting out as live, levitating in the middle of the channel, will slowly start to drop. So as we go through the time course here, you can literally see cells go from live to dead as they literally drop dead, no pun intended. Now, another interesting feature of this video is we incorporated two different stains, a calcine and a PI stain. And by imaging in both the green and red channels, what you can see is as the cells die, they go from a green to a red state. Effectively, the dye starts to permutate the cell membrane, and as the cells go lower and lower into the levitation height, you're actually seeing the stain start to report. We can look at this in a time action zone where we start with a live cell that's clearly green, and as it's slowly dying, you can see the change in its color as the PI stain becomes incorporated inside it. Now, again, the key here is that even though we are using stains to identify which cells are live, which cells are dead, these stains are only there for educational purposes. In reality, the system would behave in exactly the same fashion. So now, while we are able to get these cells to levitate at specific heights based on the live or dead separation slide, what you have to understand is that it does not matter to the platform whether you have 1 cell, 10 cells, or 100 cells that are viable and 1 million or 1 billion dead cells. Any live cell will make it into the top channel with the exact same efficiency as whether it's 1 cell or 10 million cells. This is the main difference between how our system works and any other method out there. Unlike a fax platform, which needs to see a very large number of events in order to isolate cells and then still has losses, or beads, which are just very poor at pulling cells out of suspension, even a single viable cell for us will have the same yield and recovery as 1 million cells. We've looked at multiple different cell types during the process of developing this platform. And one of the questions we asked is, well, does the performance of the system vary with the number of cells that are loaded into the system? What you can see here, we are starting with 20,000 cells, 140,000 cells, 160, 200, and 1 million cells. And what you see is the starting viability of the sample was about 25%. The post-enrichment viability is about 95% plus. What's beautiful about this image is that it demonstrates that regardless of the starting cell number, whether it's very low, 20,000, or very high, over 1 million, the performance and the ability to enrich for viable cells does not change. When we look at the yield, it also hovers right around 80%. Most of the yield issues you see here have more to do with counting. We are simply very ill-equipped to do very accurate, viable cell counts. Because you have to remember what happens is we have to collect the cells, we have to stain them, and then we have to count them. So we're looking at a 10, 20, 30-minute delay with a manual counting process to actually report how many viable cells made it out of the instrument. So the differences that you see here in the yield here have more to do with our counting issues than with the actual performance of the system. Now, this is stunning performance, and you're probably going to ask, how can it be so good? Well, if we go back to our previous in a very simple and easy to use platform, which really has a total of 3 different steps. Basically, the sample is dispensed into the cartridge. Levitation occurs within the instrument. The instrument, as I mentioned, has bright field and 2 fluorescent channels, which allows the customer to image and count the cells. And then the sample comes back out. Now, it's always nice to see how the actual levitation and imaging works. So in this particular case, I'll show you what the instrument is seeing as the sample is loaded. So as I play the video, you will see the cells flow in. What you notice is the meniscus stops before the splitter. So right here at the end of the channel, we see the splitter that was shown in the graphics before. This image is about 2 millimeters high. And what you can see is that as the sample flows in again, the meniscus stops. Now at this point, the entire sample is under the magnet and the cells are separating. What this means is that unlike any other platform, you are going to process 100% of the volume. So in the entire volume of the sample is going to be separated and collected. There are no events or cells being lost to calibration or separation. Another thing that you may ask is, well, how come some of these particular beads down here are not at the same height as the beads up here? Well, what you're actually seeing is a side product of our technology, which is we're extremely well suited to give you very good QC matrix on your beads. Any of these beads that you see here basically have a higher density than the average population that you see up here. In the next slide, we'll then collect the sample. So the sample has now been levitated and is ready for collection. The user has full control over which fraction they choose to isolate. Basically, there's a line they can move up and down. And let's say we would like to do a perfect 50 split between top and bottom. By selecting the middle level and clicking collect, the sample will then be sucked in or pulled through and collected into the separate 2 chambers we described before. So as I play this, you will see the sample go through. The splitter is right here. The cells are collected top and bottom to the 2 different collection ports. We go finish all the cells, and at the end, you see the meniscus from the air behind the volume. And again, what you're seeing here is 100% of the volume and all the cells have now been collected. And this really is what leads to our superior technical performance. So let's jump into some examples. For simplicity's sake, we'll start with some very simple cell types, THP1s and PBMCs. And again, this is run-to-run reproducibility. We're starting from about 30% viability and about 40% viability for PMCs, and are able to enrich to almost 90%. And again, as in the previous example, the yield is variable, and is variable because we are manually staining and counting the cells for collection, which means there is quite a bit of error in terms of manually counting and staining. I also mentioned that we'll be doing some comparison to the Miltenyi workflows. Here we've actually switched to primary samples, which really are the goal for most researchers. And what you see, not only is our yield superior to Miltenyi, what's really important is to think about what happens in a real world example. Here we're starting with a bladder tumor sample, and there's only 200,000 cells in this original sample. The yields for Miltenyi mean that you may end up on average with about 1,200 viable cells post-processing versus over 20,000 cells for the LeviCell. That has huge consequences in terms of what the sample can be used for. 1,200 cells is not sufficient for single cell sequencing. It is not sufficient for most workflows. Over 20,000 cells, in this case even 40,000 cells for one of the runs, is more than enough to proceed down most workflows. So it's not only that our platform is faster and easier to use, is that it is truly enabling in terms of how many cells it generates and how those cells can be used. To follow up with a few examples, here's a nice graph of a PBMC enrichment, starting at a very low viability of 20%, ending up at almost 100% viability and 90% yield. A THP1 example about the similar performance metrics, and finally a Jurkat cell example. Now obviously the real proof of the technology is in how customers are using it, and I'll show you 2 very specific examples. The first example is from the Desai lab at Stanford, and this is work where they are isolating alveolar type 2 cells. So alveolar type 2 cells are a very interesting cell type. They are very involved in both oncology and immunology. These cells tend to be very sensitive. In fact, for human alveolar type 2 cells, there are no known solutions for sorting them directly from tissue and or keeping them viable enough for culture. What the Desai lab does is they start with a healthy lung tissue, they dissociate it very gently, and then by flowing them through our system, they're able to enrich for alveolar type 2 cells, removing all the contaminants, basal cells, dead cells, and any other debris. This allows them to then get those cells into culture and use them for a variety of downstream assays. Now, what makes this example so relevant to today's work is that SARS tends to replicate in alveolar type 2 cells, so this is a very current and very important cell type to be researching. Now, obviously, given today's methods, as far as we are aware, we are the only platform that can start off with a human tissue and produce viable alveolar type 2 cells, which can then be used for downstream assays. Next example that we're going to use is an experiment that was done by a customer who's an immunotherapy company. It's based around macrophage enrichment. Now, macrophages are a very cornerstone of immune response, and as more and more is learned about macrophages, it's also important to point out that they are now a cornerstone of immunotherapy research. The ratio of macrophages present in a patient's tumor is directly correlated to that patient's survival, and macrophages themselves are present in three different states or three different polarities. One is an inactivated M0 state or naive, and then we have two M1 and M2 activated states. One is cancer killing and one is cancer protecting. So I'm sure from this very simple example, you can understand how it's critically important to be able to not only isolate viable cells, but also keep them in the original activation state. So what this experiment shows is our ability to isolate a viable live macrophage population directly from a tumor. These are solid tumors with great yield and recoveries. But even more importantly, what it shows is that the activation state as measured by gene expression of two specific genes, one specific to M1 and one specific to M2, is not altered by our processing. As far as we are aware, no other technologies out there right now allows you to obtain fully viable macrophage populations directly from tumor samples without changing or at least modifying their expression profile and in fact their activation state. Now, similar work has been done by a variety of other customers for very difficult cell types similar to the last two I showed you. These include cardiomyocytes for stem cell therapy, adipocytes for diabetes research, pancreatic cells, neurons, and so forth. And for all of these, we compare our performance to that of a FACS platform. And what you can see for the macrophage example, if you were to use FACS, your recovery and viability are subpar. You would not be able to use those cells. And we've done the same validation across many other cell types. Now, our customers speak for our technology. We have a huge number of customers who are Levitation converts. And what they really point out to us is that the beauty of the platform lies in three different things. It is simple and efficient, and because of that, it enables them to collect undamaged and unlabeled cells. And as a result, this means high yield and high viabilities, which directly enable downstream analysis and different use cases. And so with that, in summary, I'll leave you why would you why would you use the LeviCell? And the reasons are several. It's completely label-free, gives you the highest yields and viabilities possible. It's extremely easy to use because it includes debris removal and all pre-processing. And when you actually look at your true cost of obtaining a primary sample, processing that primary sample, and being able to get the most viable cells out of that sample, it is by far the lowest cost method available. So with that, we'll end.